I've been saying this for years, but size isn't everything. The cerebellum is a great example. Although it's only 10% of our brain mass, it contains over 80% of all neurons in the brain. From this, we should conclude that the cerebellum does something pretty important, and indeed it does. The cerebellum is what coordinates our movements, making sure we don't have any herky-jerky and uncoordinated actions. Instead, allowing us to move smoothly and do some pretty complicated things, like speak clearly, write our names, or juggle chainsaws. We can do any of these things only through the motor learning that occurs in the cerebellum. Of course, if you're going to do something big, like coordinate the movements of an organism, you're going to need to divide and conquer. And in the first part, we'll go over the functional divisions of the cerebellum. After that, we'll go through the circuitry in the cerebellar cortex that allows it to refine movements. The whole job of the cerebellum is to facilitate the selection of appropriate upper motor neurons so that the correct lower motor neurons can be activated and create smooth, coordinated movements. In order to do this, the cerebellum must know what we're planning, so it has to get input from the cortex. And the cerebellum also has to know what's going on in the body, so it needs input from those spinocerebellar tracts, as well as some brainstem input from the reticular formation and the vestibular nuclei. If we take a look at the cerebellum here, we can see that although it's quite small, it's also quite wrinkly. The wrinkles are there to increase surface area because all of the magic happens in the cortex of the cerebellum. And by increasing the surface area, it increases the amount of cortex, allowing it to jam-pack in there about 70 billion neurons. Current estimates place the whole brain as having somewhere about 85 billion neurons. So you can see the cerebellum contains most of the neurons in our brains. Please note its position here. The cerebellum sits just behind the pons. The pons and cerebellum both develop from the metencephalon. So, of course, they are intimately connected, and those connections are by cerebellar peduncles. Now, the cerebellum, if we take a look from the back, has a very clear midline structure here that is the vermis. And hanging off of the sides, it has a couple of hemispheres. The vermis is there to control the midline, and the hemispheres are there to control the limbs. This should make good sense, as our body is the midline, and our limbs are coming off the midline. I know this isn't anatomically correct, but none of our maps in the brain are anatomically correct. So for the vermis, think of it controlling the trunk and giving us a very good sense of balance. The hemispheres are there to control the limbs. Also assisting in balance is the flocculonodular lobe. The flocculonodular lobe is buried deep within the cerebellum, and now we can finally see some bits of it. Here we can see the flocculus, and this will run in the background, kind of a C-shaped structure here. The nodulus is also called the inferior vermis, but there's our flocculonodular lobe. You just can't see most of it because it's buried in all the wrinkles of the cerebellum. Now the cerebellum, if it's going to do its job, has to get some input. And of course it has to communicate with the rest of the nervous system. And that's accomplished by the cerebellar peduncles. There are 3 types, which we will oversimplify just a bit here. Superior, for this, you should be thinking up. We're talking to things above us. This is the output of the cerebellum. It's providing feedback mostly to the thalamus, so it can tell the cortex what to do. The middle cerebellar peduncle is the biggest, and this contains all the input from the pons. The pons gets its input from the cortex, so that middle cerebellar peduncle is the corticopontine input. In other words, it's all of the motor plans. And the inferior cerebellar peduncle is mostly input. There's a little bit of output, sure, but let's oversimplify this. Inferior, we should be thinking below. Therefore, this should be all the input coming in from the spinal cord. Let's go back just a bit. This should make good sense. Input from the spinal cord is coming from below. Input from the pons must be from the middle. And our output to tell the cortex what the heck to do is going to go up through the superior. Yes, there are exceptions, of course, but let's remember it this way just to make our lives easier. We can divide the cerebellum up anatomically, as we've done with the vermis, flocculonodular lobe, and hemispheres, and we can also divide the cerebellum up functionally. That's what we're doing here. The vestibulocerebellum is the flocculonodular lobe, as well as some other more inferior parts of the vermis. So the flocculonodular lobe is shown here, and there's a little bit of inferior vermis hanging around. But the bulk of the vermis and the hemispheres immediately adjacent to it are part of the spinocerebellum. The spinal cerebellum, as a result, will help us control our trunk and limbs. The spinal cerebellum is important for coordinating the current activity, so this will help regulate the spinal output. This will create anticipatory movements that'll prevent us from falling over, things of that nature. And the bulk of the cerebellum is a part of the cerebrocerebellum, that is the lateral portions of the hemispheres. This will still help us control our limbs, but it also does some more important stuff like planning out future actions and coordinating our speech. So there's another way of dividing up the cerebellum. Now on the bottom, down here, we've got ourselves a few different maps. So yes, we have a place for our head and arms and legs, and there's an order to it. There's also input from visual and auditory cortices, so we know what we see and what we hear, and that can influence the activity of the cerebellum. Couple weird things is that the cerebellum is organized ipsilaterally, so the right side of the cerebellum controls the right side of the body, and the left side of the cerebellum controls the left side of the body. And the motor map, you'll see the anterior lobe is purely sensory in nature. So we're putting sensory stuff in front of motor stuff. That's weird. Normally, we put motor stuff anterior to sensory stuff, right? Like in the cortex. Our primary motor cortex is in front of the primary somatosensory cortex. But in the cerebellum, the anterior lobe is sensory, while the posterior lobe has more motor functions. You can imagine all the wrinkles here. So our cerebellum's a little different. Sensory's in front of motor, and we're also ipsilateral, not contralateral. That's what we see in the cortex. In case you prefer this in table format, there you go. I'll let you read this in your own time, but here you can see how the anatomical components in the first row are divided into the different functional zones. What you should note here is the similarity between the input and the targets of the cerebellum. The vestibulocerebellum, for example, all the input is vestibular, and who are we targeting? Vestibular stuff, right? The vestibulospinal tracts. We're targeting the oculomotor nuclei, right, to control the eyes. The spinocerebellum, well, it gets input from the spinal cord, reticular formation. The trigeminal nerve also contributes a bit. We do know some planned action as well. And who are we targeting? Well, all of those spinal tracts, right? We get input from the spinal cord. We provide output to the spinal cord. We'll also hit the cortex by hitting the thalamus as well. That's that ventrolateral nucleus of the thalamus there. Hey, the cerebrocerebellum. All of our input is from the cortex via the pons. Mostly what we're targeting, ventrolateral thalamus, which then targets the cortex. So whoever's talking to us, that's who we talk to in the cerebellum. And that allows the different functional zones to have different functions. So the vestibulocerebellum is really important for coordinating the vestibulo-ocular reflex and some postural movements to prevent us from falling over. The spinocerebellum produces anticipatory limb movements and also coordinates the activation of antagonist muscles to smoothly terminate movements. No herky-jerky movements here. The cerebrocerebellum plays a key role in motor planning as well as motor learning, which will come up in the second part of this talk. And in case you like to see something 3 times, here are the functional zones now in a nice, easy to digest flowchart. Spend some time with this and make sure it makes sense. Now, the outputs of the cerebellum are from the deep nuclei. These are not in the cortex. That's why they're called deep nuclei. They are buried deep in the white matter. The fastigial nucleus, which is used by the spinocerebellum, targets the vestibular nuclei and reticular formation. As we can see here, 2 targets, and these 2 will create the reticulospinal tracts and vestibulospinal tracts, helping us to coordinate movements automatically. We do provide a bit of descending feedback to the cortex, but the major output here we should be thinking is the descending output. The interposed nuclei, which is the globose and emboliform nuclei, mostly we're providing upward feedback, so we're projecting upward. 2 locations here. We're hitting the red nucleus to activate the rubrospinal tract. In this case, it's upper limb flexors. And we'll hit the ventrolateral nucleus to target the motor cortex and the association cortices. And this is all with motor coordination. So we can help coordinate the activity of upper motor neurons based on sensory input. Because keep in mind, the interposed nuclei are a part of the spinocerebellum. The cerebrocerebellum uses the dentate nucleus, and this is the largest of the deep nuclei. This one, mostly we're targeting the ventrolateral thalamus, and in this case, we're going to play a role then in motor coordination. So which upper motor neurons are we going to activate? In this case, the dentate gets mostly its input from the corticopontocerebellar input. So now we're coordinating action based on what we plan to do. Based on our years of motor learning, we've learned how to properly do whatever it is we're talking about. If it's in the lab, we're probably touching our nose with our finger and then someone else's finger with it. In real life, it's everything that you do. The other target is the red nucleus, but notice, not the same part. Instead, we're going to hit some smaller neurons within the red nucleus that don't create the rubrospinal tract. Instead, they activate the inferior olive, and this is key for motor learning. More on this later. So the whole function of the cerebellum is the coordination of our voluntary movements as well as some involuntary movements to help smooth everything out. So we have a good idea of what's going on. Here we can see the descending input. So the cortico-ponto-cerebellar input. What are our plans? What do we want to do? We also have sensory input. What's actually going on in our body? So where is my body? And what do I want to do with it? If I know those two things, assuming I've learned how to do the action, the cerebellum can tell us how. A couple things that it's going to do here. It can create some non-conscious anticipatory movements. For example, if you're going to pick up heavy weight or something like that, in this case, if we're going to do a little biceps curl, well, that's going to apply force under the body and could very well cause us to lean forward, especially if we're pulling on something that's attached to the wall here. In real life, maybe we're just picking up a barbell. Well, before we do that, before we actually curl the biceps, notice earlier than that, we see activation of the leg muscles. In other words, we start bearing weight, anticipating that forward movement that comes from the curl. Of course, we don't think about this. The only thing this guy's thinking is curl. But his cerebellum is thinking, don't fall over. So sure, we'll have some flexion at the elbow, but we're also going to recruit those lower limb muscles to make sure we don't fall over. Note the term here, bulbospinal. That just means from the brainstem to the spinal cord. So in this case, it's the vestibulospinal tracts and the reticulospinal tracts that we can recruit. The other thing that the cerebellum will do is refine our movements. So we have a plan based on what we've learned. Let's adjust which upper motor neurons we activate and in which order and with what timing. This is the function of the spino and cerebellum. And this is all about that feedback to the thalamus, telling us which neurons in the primary motor cortex to activate and in what order by going to the premotor cortex. For example, if we want to smoothly terminate a movement, we need not just stop the movement. That's not going to work. So let's say we're talking about extension of the elbow. At the end of that movement, it's not just turning off the extensor, it's actually stimulating the flexor, then letting both of them off together. So we have to coordinate the activity of both the agonist and antagonist muscle. If you just stop applying if you just stop applying extension, that causes the elastic components of the muscle to create, tremor. There we go. So when we have damage to the cerebellum, we'll see intention tremor because we can't possibly coordinate agonist and antagonist movements. When we can, though, this ensures that we don't have tremor. We can smoothly stop our movements. So when we have damage to the cerebellum, it's quite easy to see what problems arise. When we have damage to the vermis, that's going to affect the trunk and thus affect balance, and we'll get truncal ataxia. In this case, we have severely impaired balance, where we can have difficulty sitting in more extreme cases, certainly some difficulty standing. And notice this gentleman's drunken gait, which is simply a way of describing a wide-based and unsteady gait. Truncal ataxia is fairly easy to see with any sort of gait testing or the Romberg test, where subjects stand still with their eyes closed and hopefully don't sway very much. The appendicular ataxia occurs when we have damage to the hemispheres. And note it's ipsilateral. A common way of testing this is the finger-nose-finger or the heel-shin tests for the upper and lower limbs, respectively. Now notice, right limb totally fine. Left limb, notice the tremor. We're having problems with that agonist-antagonist coordination there. So the elastic components of the muscle are making it very hard to put his finger where he wants it. Also, that lack of coordination makes it very hard for him to put his finger on his nose. And notice little tremor there. That intention tremor is characteristic of damage to the cerebellum. This ataxia is characterized by problems moving the correct distance and with the correct timing. That is dysmetria and dysrhythmia, respectively. We also see problems with fast alternating movements, and this is called dysdiadochokinesia. There are additional symptoms that arise besides problems with the trunk and limbs. Eye movements are impaired. The type of impairment depends on where the cerebellum is damaged. We can have issues with the speed of our saccades if we have damage to the superior vermis. But more inferior portions, such as those found in the vestibulocerebellum, create problems with smooth pursuit than the vestibulo-ocular reflex. Both of those are controlled by the vestibular nuclei. What we can see here is that the speed of the vestibulo-ocular reflex is all thrown off. The gray line is showing movement of the head. Now, ideally, the black line there, the movement of the eyes, should match up perfectly. But notice, it's a little slow to get activated down here, and when it is activated, it has an inappropriate rate. It's too fast. And that's because of damage to the cerebellum. So the speed of his vestibulo-ocular reflex is thrown off. As a result, his eyes get too far off track, and he has a corrective movement back to where it should be. Again, a little too fast, and we'll have to correct again. Damage to the cerebellum can affect activity in the vestibular nuclei, creating inappropriate activity there, and that is what accounts for the nausea and vomiting. This is essentially motion sickness without any motion necessary. So when the vestibular nuclei have activity that doesn't match up with visual perceptions, this causes us to vomit. Not sure what the rationale is there. It probably has something to do with the medulla, monitoring the blood to look for toxins, but certainly the case that when we have inappropriate vestibular activity, that makes us feel ill. So damage in the cerebellum creates inappropriate vestibular activity. That throws off not only the VOR, but also creates that nausea. Now here we can see some gaze-evoked nystagmus. So she's having a problem putting her eyes where they should be. She wants to hold them now to the right, but they keep drifting inappropriately away. That's why we get the gaze-evoked nystagmus. Let's have a look at this again. So notice the eyes are flickering there. They keep drifting away, and she has to have that corrective nystagmus to hold them back in place. This is characteristic of damage to the flocculonodular lobe or the vestibulocerebellum. Additional signs include those of speech, where we have slurred or scanning speech, also called staccato speech. So each word is broken into each syllable. The slurring of speech, also called dysarthria, can occur with damage to the cerebellum as well. Let's hear an example.
Today is a nice day. Today is a nice day. Today is a nice day.
In case the quality of that sucks, you can see the link up above to check it out in your own time. Now, if we have damage that's in the cerebellum, there are some additional signs we won't see. There should be no change in our strength. There should be no change in reflexes, and there should be no change in conscious proprioception. We will certainly have changes in non-conscious proprioception. In other words, the cerebellum won't know where the body is, and thus won't be able to properly coordinate our movements. So let's go through some of the magic of the cerebellum. And what we're going to find here is that mostly it's a bunch of inhibition. There's only one excitatory neuron that lives in the cerebellar cortex. That's going to make our life easier. This will create a very simple loop through the cerebellar cortex. And if our movements don't work out in our favor, input from the olive can erase that pattern. All right, so we got 3 layers, 5 cells. That's not so bad. On the outside, we have the molecular layer, and it's called that because it's mostly made of little tiny things. Those little tiny things are the little red dots shown up here, the parallel fibers. So those run in the molecular layer. They're really small. That's why it's called the molecular layer. Also found living around there, we've got the stellate and basket cells. These are both inhibitory neurons, and they're going to inhibit the Purkinje cells. More on this later. Purkinje cells are also inhibitory neurons. These are the neurons that inhibit the output of the cerebellum. Thus, they're the ones that provide the feedback to our motor plans. The granule cell layer contains 2 types of neurons. The granule cells, hence the name. These are very, very small. That's how they got their name, granule cells. Here they are, shown in red. These comprise about 75% of all the neurons in our brain, and they're the only glutamatergic neuron in the cerebellar cortex. Everything else is GABAergic. We have one last GABAergic cell, the Golgi cell, and that inhibits the granule cell. So the granule cells are the input. This is the first stop in the cerebellar cortex. Now, the input into the cortex are collectively called mossy fibers. These are from a variety of sources. It could be from that corticopontocerebellar input. It might be the spinocerebellar input or vestibulocerebellar input. As long as it's not from the olive, it's a mossy fiber. These are excitatory, and they're going to excite a couple of things. First, they're going to excite the deep cerebellar nuclei. So they'll provide a rough draft to the output. The other target, the granule cell. So mossy fibers excite granule cells. Granule cells, when excited, also release glutamate. Not on mossy fibers, they use the parallel fibers. So the schematic here, mossy fiber stimulates granule cell, which then creates a parallel fiber. And you know what? Maybe, hold on. Okay, there we go. Sorry for the clumsiness. My computer needs a cerebellum. Alrighty. Let's keep track of this stuff. So we got mossy fibers. And that's basically everything. Except the inferior olive. Those are their own type of fiber. Mossy fibers give us input to the deep nuclei. Remember, this is the output. Dentate projects upward. Vestigial nucleus projects downward. You remember this. This is our rough draft. Within the cerebellar cortex, we target the granule cells. Granule cells then create parallel fibers that run within the molecular layer. Well, let's label that as the cerebellar cortex. So the deep nuclei are not in the cerebellar cortex. Alrighty. Parallel fibers run perpendicular to the Purkinje cell dendrites. Purkinje cells make these really elaborate but flat dendritic arbors, and as the parallel fibers pass through, they'll make one or a couple synapses, but not that many. Let's expand our schematic here now to include a Purkinje cell. This, of course, has massive dendritic arbor here, which I'll keep a little simple. So Purkinje cells have their cell bodies in the Purkinje cell layer. The granule cell layer has the granule cells, and the molecular layer has our parallel fibers. Alrighty, there's our current schematic. Now, before we exit the granule cell layer, we should keep in mind that we have Golgi cells, and their whole purpose is to filter out weakly activated granule cells. Golgi cells get input from granule cells, and they also get input from Purkinje cells. Granule cells excite them, Purkinje cells inhibit them. Golgi cells, if active, inhibit granule cells. This creates a nice little filter. So let's say we have strong input. Mossy fibers strongly stimulate the granule cell, and its activity actually turns on Purkinje cells. Those Purkinje cells prevent the Golgi cell from inhibiting the granule cell. In other words, maintaining its activity. However, if we don't have very strong input, then granule cells that are weakly activated probably don't contribute to Purkinje cell activity, and thus the Golgi cell can inhibit the granule cell. The whole point of this is to not waste energy. Only granule cells that activate Purkinje cells remain active. Granule cells that do not activate Purkinje cells are thus inhibited. So unless you're contributing to the output of the cerebellum, there's no need for you to be active. Purkinje cells then target the deep nuclei. Still not there. Okay. So, little offshoot here. The Golgi neuron targeted by Purkinje cells. But the main thing is this right here. These are the edits that the cerebellum provides to the rough draft of the mossy fibers. It's inhibition from Purkinje cells to the deep nuclei. And remember, deep nuclei are the output. They project out those peduncles. So Purkinje cells get a whole heck of a lot of input from parallel fibers, as you can see there, and they have one climbing fiber that strongly excites them. This is our last fiber to think about. So we had mossy fibers, we got parallel fibers, and now we have to consider climbing fibers from the inferior olive. Let's go back to our schematic here. So everything except the inferior olive would be the mossy fibers. The inferior olive, which is critical for motor learning, as mentioned earlier, projects into the cerebellum and strongly targets a single Purkinje cell. That is the climbing fiber. And it's called the climbing fiber because it looks like it climbs along the dendrites, forming many synapses onto the same Purkinje cell. So many synapses with few Purkinje cells. Parallel fibers, on the other hand, form very few synapses, but with many Purkinje cells. Purkinje cells then provide GABAergic inhibition to Golgi cells, as shown before, and to the deep nuclei, thus affecting cerebellar output. And there we have it, a very simple little loop where we get input from the mossy fibers, exciting granule cells. A large number of granule cells can collectively excite Purkinje cells that then inhibit the deep nuclei. Now we also have some inhibitory neurons in that molecular layer, and we'll just lump them together and call them, we'll lump them together in our schematic. We have different names for them. Stellate cells target the dendrites of Purkinje cells. Basket cells form what looks like a basket of inhibitory synapses around the cell body of the Purkinje cell. But both of these do the same thing. They inhibit Purkinje cells. So just to get these mixed in here, stellate cells, which kind of look like a star, inhibit the dendrites, and basket cells inhibit the cell body, forming kind of a basket of inhibitory synapses. All right, what turns them on? So, of course, parallel fibers. And their purpose is to provide lateral inhibition, to make sure only the Purkinje cells that are excited get to remain active. Okay, so let's say we have input from the middle parallel fibers there. We're going to turn on the Purkinje cell number 2, and then by turning on the nearby basket and stellate cells, we turn off the neighbors. Let's go back and see without. Notice they're all this middling shade of gray. Purkinje cell 2 will get brighter. That is more active. I can see that I've screwed my label up there. You know what? Hell, I think I can fix that right now. What's wrong with me? Okay, look at that. On the fly, fixing things. All right, so I'm a moron. Yes, I will fix this right in front of you. No, I will not edit this out. Alrighty, so now, finally, the dummy behind the screen here has fixed it. So now when I strongly excite Purkinje cell 2, I inhibit Purkinje cells 1 and 3. And this makes Purkinje cell 2 pop. It looks more active than its neighbors. So this creates a very clearer pattern in the cerebellum. That's all lateral inhibition is. Everywhere we see it, it has the exact same function. Clean up the noise. If Purkinje cells 1 and 3 are not excited, don't let them be active. Because here's the deal. Purkinje cells are spontaneously active. So we need to silence the noise. That's the whole purpose of lateral inhibition. As you can see here, this Purkinje cell is indeed spontaneously active. It's firing away. So the lateral inhibition will help clean that up. What climbing fibers do is provide the means for motor learning. So climbing fibers, also called the olivo-cerebellar tract, strongly excite individual Purkinje cells, as we can see here. So there's the orange climbing fiber, and it creates this nice complex spike. It's called that because the Purkinje cell is so excited you can't tell the individual spikes apart. The climbing fiber forms many, many, many synapses, which are all excitatory and all active at the same time. So that Purkinje cell is strongly excited and fills up with calcium. What that does is stimulate long-term depression. But we only want to weaken synapses that were active. And the mechanism for detecting this is shown right here. So long-term depression really just means a long-term reduction in synapse strength. That's it. It has nothing to do with mood. We're just decreasing synapse strength. So when you see long-term depression or LTD, you should just think decrease in synapse strength. Okay, the climbing fiber is there to provide strong depolarization and fill the cell up with calcium. Calcium can diffuse, and it goes into all of the synapses, even the ones that weren't active. So calcium is nonspecific. Everywhere has calcium. Notice we got a global increase in calcium. This is one of the things I need to activate protein kinase C. This one is the star. The other thing I need is diacylglycerol. That's this thing right here. If I have these two things, 1 plus 2 equals protein kinase C activation. I only have diacylglycerol floating around in the membrane if I recently received glutamate from a parallel fiber. Glutamate will activate AMPA receptors to provide depolarization, but it also activates metabotropic glutamate receptors. mGluR1 is GQ coupled. That means it activates phospholipase C, which then cuts up phosphatidylinositol bisphosphate to make diacylglycerol. Excellent. So only at those active synapses do I have diacylglycerol. What protein kinase C does is phosphorylate AMPA receptors, causing them to be removed from the surface. All right, so what does that look like? Ha, look at that. Went the right way. Okay, let's imagine two synapses. Here's my parallel fibers. They're running in parallel with one another. This one was active and dumped out some glutamate, which of course was in a vesicle. Yikes. 7 transmembrane domains on this metabotropic glutamate receptor. But of course, bread and butter excitation is through the AMPA receptor. Over here, I got the same setup, but no glutamate. GQ activates phospholipase C, which then makes diacylglycerol in the membrane and some calcium. But of course, we had calcium from the climbing fiber. Right, the climbing fiber input is very strong. That triggers the complex spike. And so the whole cell fills up with calcium. Calcium's everywhere. Has some calcium here, has some calcium here. In order to activate protein kinase C, though, I need two things, diacylglycerol and calcium. I only have that at synapse A over here, not synapse B. So only in synapse A do I get activation of protein kinase C. Over here, not so much. That's actually a good thing, though, because what protein kinase C is going to do is get rid of AMPA receptors. So over here, let me just clean up a little bit of this mess. We appreciate it's GQ coupled, right? Calcium plus diacylglycerol activate protein kinase C, and protein kinase C causes internalization of AMPA receptors. In other words, we remove it from the surface. Now, this parallel fiber will no longer be able to excite this given Purkinje cell because its synapse is weakened. Instead, a different parallel fiber might be able to communicate. So notice what's happening with motor learning. It's just long-term depression. That is the removal of AMPA receptors. All we're doing is telling the cerebellum what not to do. We're never actually telling it what to do. That's why we have to try things over and over and over and over until we finally get it right. Then, without the error signal, we don't trigger long-term depression, and we continue to practice. Instead, long-term potentiation strengthens synapses. And we'll talk about that in later lectures. So here's our working model. This is in Word format. I'll have some pictures for you here. But the idea is that the cerebellum gets input from mossy fibers. All that input from what we're planning, what's going on in the body, that gets integrated through the parallel fibers onto Purkinje cells. And Purkinje cells then modify the deep nuclei output. Deep nuclei project to different motor areas to ultimately shape the activity of upper motor neurons. If we have an error, that's what's on the bottom here, we're going to get a little LTD via the climbing fibers, so we don't provide that same pattern of feedback. Here it is in a little flow chart format. I've modified just the parts that are blue. But we've got our mossy fiber input. The rough draft goes right to the deep nuclei. We also go through the cerebellar cortex. Granule cells, parallel fibers activate Purkinje cells, and Purkinje cells then inhibit the deep nuclei, and that would be the cerebellar edits. So mossy fibers give the rough draft, Purkinje cells give the edits. The deep nuclei create some kind of motor output. If it works, that's great, keep it up. If it doesn't work, let's say we fall down or we don't put the ball where we want it, that failure stimulates the inferior olive, and climbing fibers modify the connection between granule cells and Purkinje cells. So the next time we have that same given input, let's say I'm on the free throw line and I want to put the ball through the hole. Well, what I tried last time didn't work. Now I'll try something different. If that pattern of edits works out and the ball goes in the hole, great, try that again. If the ball does not go in the hole, failure, erase it, try again. Again, I don't know what to do, just don't do that same thing. Here it is, simplified just a little bit. So we got mossy fiber input providing our rough draft to the deep nuclei. And here I'm just saying there's a certain pattern of motor output. We'll chop the deep nuclei into just 3 divisions, and they're all equally active, divisions A, B, and C. We also, via the mossy fibers, excite granule cells. There's a bunch of these, and they'll go talk to hundreds of thousands of Purkinje cells, and let's say these 3 were active, and they'll provide equal inhibition to the 3 segments of the deep nuclei. So motor output is kind of at a moderate level for all 3 of my deep nuclei zones. Let's say that didn't work. That didn't get the ball through the hole. Okay, the inferior olive is going to come in, strongly excite one of these Purkinje cells and remove that from the pattern. Now that mossy fiber input still creates the original rough draft, but I have modified feedback. So now I'm going to change the pattern of motor output, and hey, maybe this one works. And that is our summary of the cerebellum. If you have any questions, put them in the questions box. If you can't answer any of these questions, think a little bit harder. If that doesn't work out for you, obviously come by my office or use the questions box to your advantage. See you later.